Today in this module, we are going to talk about bioremediation. We all know that Earth is a beautiful place to live in, with beautiful nature around. But in course of time, because of construction sites, industrialization, advancement in technologies, and basically because of human interference, nature is getting polluted. Now to eradicate this pollution, bioremediation comes into picture. Bioremediation is a waste management technique which involves the use of biological organisms to remove or neutralize the pollutants from a contaminated site. So in this module, we will be learning what is bioremediation, the types of bioremediation, its advances and the disadvantages of bioremediation. Bioremediation is a waste management technique that involves the use of organisms to remove or neutralize pollutants from a contaminated site. Bioremediation is a treatment that uses naturally occurring organisms to break down hazardous substances into less toxic or non-toxic substances. Technologies can be generally classified as in situ or ex situ. In situ bioremediation involves treating the contaminated material at the site, while ex situ involves the removal of contaminated material to be treated elsewhere. Recent advancements have also proven successful via the addition of matched microbe strains to the medium to enhance the resident microbe population's ability to break down contaminants. Microorganisms used to perform the function of bioremediation are known as bioremediators. Bioremediation can occur on its own in nature or can be spurred via addition of fertilizers for the enhancement of bioavailability within the medium. Bioventing, bioleaching, bioreactor, bioaugmentation, composting, biostimulation, land farming, phytoremediation and rhizofiltration are all examples of bioremediation technologies. On the basis of removal and transportation of waste, bioremediation technology can be classified as in situ and ex situ. In situ bioremediation involves treatment of contaminated material at the same site, while ex situ involves complete removal of the contaminant material from one site and its transfer to another site, where it has been treated using biological agents. In the comparison of both methods, it was found that the rate of biodegradation and consistency of process outcome differs between in situ and ex situ methods. With the need for excavation of contaminated samples for treatment, the cost of ex situ bioremediation is relatively high as compared to in situ. In situ and ex situ, both the bioremediation methods depends essentially on microbial metabolism. However, so far in situ methods are preferred over ex situ for ecological restoration of contaminated soil, water and environment. Types of bioremediation. Phytoremediation. The three main steps in inorganic ion transport in the simplest pathways are active transport of metals across root membranes, entry of metals into symplast during translocation from root to shoot, and chelation and sequestration of metals into specific compartments in the leaves. Phytoremediation, the use of plants to remove or degrade contamination from soils and surface waters, has been proposed as a cheap, sustainable, effective and environmentally friendly alternative to conventional remediation technologies. Plants use solar energy to extract chemicals from the soil and to deposit them in the above ground part of their bodies or to convert them to a less toxic form. These plants can then be harvested and treated, removing the pollutants. An ideal bioremediator would have high tolerance to the pollutant, the ability to either degrade or concentrate the contaminant at high levels in the biomass, extensive root systems, the capacity to absorb large amounts of water from the soil, and fast growth rates and high levels of biomass. Phytoremediation of heavy metals from the environment serves as an excellent example of the process of plant facilitated bioremediation and its role in removing environmental stress. The ideal type of phytoremediator is a species that creates a large biomass, grows quickly and has extensive root system and must be easily cultivated and harvested. For example, the plants like Thalassae and Brassica gensea. Now let's talk about the mechanism of phytoremediation. The entry of heavy metals into the plant system is a complex process and involves both the apoplastic and symplastic pathways. We had talked about the three main steps in inorganic ion transport in the symplastic pathways, that is the active transport of metals across the root membranes, entry of metals into symplast during translocation from root to shoot, and chelation and sequestration of metals into specific compartments in the leaves. Chelation of metals within the plant allows the xylem loading and transport as well as for sequestration. These mechanisms involve many metal specific chelators, that is ligands and organic acids, many of which have only begun to be studied or have not yet been characterized. The role of chelators in hyperaccumulation is to form complexes with heavy ions. This can serve the function of aiding in transport, or it can be the terminus of the ion leading to sequestration in the shoot of the plant. Metallothioneins and phytochelatins are two classes of chelators involved in metal accumulation. Different organic acids and ligands have been found associated with various metals in distinct parts of different plants. For example, in Thalassae, most zinc in roots was associated with histidine, while in shoots it was associated with organic acids. In Thalassae, cadmium in the leaves was found to be bound with sulfur ligands. In Arabidopsis, zinc was mostly stored in the vacuoles of mesophils, while in Thalassae it was in the vacuoles of epidermal cells. There are several drawbacks and the advantages of phytoremediations. There are several advantages of phytoextraction. The cost of phytoextraction is fairly low when compared to conventional methods. Another benefit is that contaminant is permanently removed from the soil. In addition, the amount of waste material that must be disposed of is substantially decreased, and in some cases the contaminant can be recycled from the contaminated plant biomass. The use of hyperaccumulator species is limited by slow growth, shallow root system and small biomass production. There are several factors limiting the extent of metal phytoextraction, including metal bioavailability within the rhizosphere, rate of metal uptake by roots, proportion of metals within the roots, rate of xylem, loading and translocation to shoots, cellular tolerance to toxic metals. The drawbacks of phytoremediation. Depending on the growing conditions required by the plant, the phytoremediation process is dependent on it. Tolerance of the plant to the pollutant affect the success for remediation. Contaminants collected in senescing tissues may be released back into the environment in certain seasons. Time taken to remediate sites far exceeds that of other technologies. Contaminant solubility may be increased, leading to greater environmental damage, and the possibility of leaching may occur. Next, we will be talking about